Boa tarde a todos e todas que nos acompanham hoje em mais um SBV Talks, né, um evento que a gente tornou tradicional e que tem tido, né, uma aderência extremamente importante por parte de virologistas, pesquisadores, estudantes. Né? Então é uma honra, um prazer estar aqui com vocês mais uma vez em mais esse evento, né, do SBV Talks. E, né, a gente agora se aproxima do nosso principal evento do ano, que é o Congresso Brasileiro de Virologia, né? Então, no mês de outubro, nós não teremos um SBV Talks, exatamente porque vai acontecer o nosso Congresso Brasileiro de Virologia. Então, esse evento, ele encerra essa primeira parte do ano. No mês que vem, nós teremos o nosso congresso. E em novembro, nós teremos a última sessão, o último evento do SBV Talks no ano de 2021. Né, em dezembro a gente faz um recesso, já que as últimas quintas-feiras, a última quinta-feira de dezembro cai num feriado, a gente não vai ter esse evento, não vai ter o SBV Talks. Em janeiro nós ainda vamos discutir se nós teremos o evento, provavelmente sim, mas a gente libera, né, a agenda de 2022 assim que nós já tivermos as confirmações das apresentações para o ano que vem. Tá? Então, eu não vou me delongar, né? Hoje a nossa atração principal, né, é a doutora Stacey Schultz-Cherry, que vai ser apresentada pela doutora Helena, que é a nossa primeira secretária da SBV. Desejo a todos, então, uma tarde muito proveitosa, né, e aproveitem bem a palestra da doutora Stacey. Helena, por favor.
Hello, good afternoon. It's a very exciting moment because we are happy to have Dr. Stacey Schultz-Cherry here today. She is a full member in the Department of Infectious Disease at St. Jude's Children's Research Hospital in Memphis, Tennessee, and where she is the co-director of the Center for Excellence in Influenza Research and Response. She also received her PhD in Molecular and Cellular Pathology from the University of Alabama, and did her postdoctoral training in influenza virology at the University of Wisconsin. Before she joined the faculty at St. Jude's Children's Research Hospital, she was a tenured associate professor in the Department of Medical Microbiology and Immunology at the University of Wisconsin-Madison. She's recognized internationally for her studies on the pathogenesis of influenza and enteric viroses in high-sick populations and influenza at the animal-human interface. She is author and co-author of over 200 research articles, reviews, book chapters, and she is past president of the American Society for Virology and currently chair of the Public and Scientific Affairs Committee for the American Society for Microbiology. So it's a great honor to have you, Dr. Stacey, here today. And let's hear about avian influenza in South America, what we know and what we do not know. Thank you very much for accepting the invitation.
No, thank you so much, Helena and Flavio. It is an absolute pleasure to be here. I'm going to share my screen, I hope. Okay. Can you see that?
Yes, we're seeing.
Yes, beautiful. Thank you so much. Well, again, thank you so much for the invitation to speak. I am honored to be here. And as you heard, my name is Stacey Schultz-Cherry. I am at St. Jude Children's Research Hospital. And I'm going to talk to you today about avian influenza viruses and really influenza virus overall in South America. So the focus of my laboratory is really understanding how your body responds to viral infections, particularly influenza virus, as well as enteric viruses like astrovirus and norovirus. We're very interested in both the epithelial and innate immune responses, how the virus fights back by evolving and changing, and then developing better therapies, vaccines or antivirals. We really focus on these viruses in vulnerable populations with with influenza, it's people that are overweight or obese or even malnourished and underweight, and more recently in pregnant women. And with our enteric viruses, we actually look in the patient population at the hospital where I work, which is a pediatric cancer or oncology hospital. So we look in immunocompromised children. You know, like many people, many virologists, I think we've been pulled into working with SARS-CoV-2, where we're also looking in these high-risk populations. As Deputy Director of a World Health Organization Collaborating Center for Influenza, we're also responsible for really going out there and looking for influenza viruses to really try and understand zoonotic transmission and doing global animal-human interface studies. And to talk more about that, many people ask, if we're at a child cancer hospital, why do we work on influenza viruses? And really the answer to that is that St. Jude has been a leader in influenza viruses for 40 plus years, because this is the home of Robert Webster, who is probably arguably one of the most well-recognized influenza researchers around the world. His work has really been instrumental in understanding the role of wild birds as a reservoir for influenza virus, how they spill over into poultry and even swine. And he's really one of the first people to start working on vaccines. Because of Dr. Webster's long history at St. Jude and with influenza, we have been well funded by the National Institutes of Health here in the United States to study influenza virus. We have a Center for Excellence in Influenza Research and Surveillance. It really began in about 1997, and we had a center from 2007 to 2012, and then again here at some of the other centers. And we just recompeted this, and it started in April. And here are the other groups that are also have centers for excellence. It's now Center of Excellence in Influenza Research and Response, because our goal is to really try and do pandemic preparedness. So these are the sites, this map shows you the sites where St. Jude has collaborations to look at influenza and other emerging viruses around the world. The red dots are where we do avian studies. This red with black is avian and ruminants for things like MERS and SARS. Pink is swine, blue is human, and then you can see yellow is SARS. We'll have to, or MERS, we'll have to update it for SARS and Zika. And we joke that anything that flies, swims, runs, if we can catch it, we're going to swab it, because we want to understand what is happening in the animal populations, which is so important for the influenza ecology, and then how those are getting into humans. And what we do is when we isolate these viruses, we really use them to understand the immune response to infection, how your body is responding, and then how is that virus transmitting, and what are the molecular properties that might allow a virus to really jump from an animal into a human. And this is something that has been more in about the last 5 years, really moving our long-term studies in wild birds and then in swine to looking at influenza at this animal-human interface. And this is, again, happening all around the world. What we can do with this information is really supply it to the World Health Organization. As I mentioned, we have a collaborating center for influenza virus. There are only 6 of those around the world, one in China, Japan, Australia, London, Atlanta, CDC, and then here in Memphis. And we're unique in that we were really the only center for a very long time to focus on those zoonotic viruses. Everybody else is human seasonal viruses. And we work with influenza collaborating and national influenza centers around the world, H5 reference laboratories, FAO, to really provide guidance and oversight on zoonotic infections and developing vaccines or pre-pandemic vaccines to viruses we find in animals that are infecting people. So this is typically in the United States the time of the year that we start really promoting people to get their influenza vaccine, which is so critically important. But as most of us know, the past really 2 seasons has been very different. It's been a very different influenza season in that we're not seeing a lot of circulation of influenza virus. So this is a map from the World Health Organization beginning of September, and it shows different colors or the different strains of flu. So influenza is still circulating. It's just at much, much lower levels. And that's really shown here on this graph, where it's the percent of visits for influenza-like illness and the week. And you can see this is a 2009 pandemic. These are all of our other years. Here is 2020, 21. So we are seeing a bit more influenza virus, at least in the US and other parts of the world, this season than we did last. But really during SARS-CoV-2 pandemic, we've seen much, much lower levels of influenza. What's happening in Brazil? Well, this is a recent, some recent data from PAHO. And what you can see here is this is influenza distribution, and it's by 2015, 16, 17. Basically, you can see really since the pandemic has started, 2020, 2021, this red flat line for influenza. And this purple shows you SARS-CoV-2. So why? Why are we seeing much less influenza virus? You know, I think there's a lot of different possibilities. One being the measures that have been put in place to stop the spread of the pandemic, of the SARS-CoV-2. Masks, less travel, quarantine measures, even things like schools being virtual rather than in person. All of those are probably limiting influenza and other respiratory virus transmission as well. An alternative hypothesis that people are testing in the laboratory is that there's some kind of competition between the viruses, in that if you have SARS-CoV-2, you can't get flu, and that's also limiting spread. But we really don't know yet what it is. I would imagine it's probably a lot of the quarantine measures. So this is what's happening in the human population. It is a very different story when you look at birds and pigs, where the virus has not only not slowed down, it has actually, we've seen increased emergence and spread of influenza viruses. You know, both avian influenza viruses, H5s, H7s, and we'll talk more about those, as well as the swine influenza viruses. So this is a nice map showing you sort of the distribution of just H5 influenza virus, and you can see many parts of the world. And then when you look at swine influenza viruses, what this is showing you the different colors is each a different strain of swine influenza virus. And it goes from December 2020 to really more frequently. And what you can see is each dot is a different strain and the amount of that virus. And you can see here from really June to August of this year, there is a huge explosion in the amount of swine influenza virus being found around the world, as well as the number of different strains of virus that are being found. And this is troubling because we know influenza has an extensive host species. So we have 4 types of influenza viruses: influenza A, B, C, and now D. And what you can see is that men can get infected and pigs can get infected with virtually all types of influenza virus. But these influenza A viruses have this really extensive host range. We know bats can be infected with influenza viruses. And we always find new species that get infected. And this becomes problematic with the influenza A's because these are the ones that can really cross species and get into man. So here we see this sort of wheel of influenza misfortune. And you've got these wild aquatic birds in the middle that are the reservoir for influenza viruses. And what you can see is that these wild birds can spread influenza to basically every other species. And then many of those can then directly or indirectly infect man. And lots of different strains that can at this point. So really any area where you have a mixing of different species, especially wild birds with domestic poultry, with mammals of many different kinds, whether they be backyard production systems, commercial production systems, live bird markets, even, backyard farms are areas where we can see transmission of influenza into people. So influenza is probably, of all the viruses, the best at crossing species barriers. And the question becomes, why is that? It's partially because of the genetics of the virus. So it's, as you know, it's a negative strand RNA virus, but it's segmented. So each of its genes is on a unique segment. And that allows a lot of mixing and matching of genes. But the other thing that is typical of a negative strand RNA virus is that its RNA-dependent RNA polymerase has no proofreading function. So flu can undergo antigenic drift in that while it's replicating, it makes errors, and it's always sort of on the verge of error catastrophe, in that there are mutations made frequently. It's usually about 1 in about 1,000 nucleotides. So that means you have very few actually infectious viral particles that come out when you do an infection. But all you really need to get is one mutation that gives that virus some type of advantage, whether it be, evading the immune response, getting to changing tropism, or crossing a species barrier. That becomes the predominant population. And that, you can see, is why we have to update the influenza vaccines so frequently, is because of this antigenic drift. And I think, most of the world now understands antigenic drift much better because of the coronavirus pandemic, unfortunately, and watching these variants emerge. So hopefully it will help people understand why we have to update the flu shots so frequently. But influenza also undergoes antigenic shift. This is where you have a species get infected with different strains of influenza virus and start mixing and matching those genome segments. And we're starting to understand more about which segments have to sort of go together. Usually HA and M work together, and you'll see those reassorted. But we really still don't understand reassorting and what makes one virus successful at reassorting, which species is the best at doing this, and how we stop this. But once you get reassorting, you've got the possibility of a virus emerging that can go back, and this is where we obviously get our pandemics. And for, I think, a very long time, the paradigm or the dogma in the field that pigs were the mixing vessel. It's always the pigs. But I think we've become much more open-minded and aware that there are many, many different possible mixing vessels. Man could be a mixing vessel, birds. We're even seeing our furry friends, our pets. Dogs seem to be a very good mixing vessel. Cats, somewhat, don't typically transmit much to humans, but dogs we definitely see are mixing. And I think now we better appreciate, because of the H7N9 virus that emerged in China, which we'll talk about, that even birds can be a mixing vessel that lead to a virus that can then infect man. So let's talk more about avian influenza viruses in people, and what do we know? So this is a really nice timeline of the avian influenza viruses since they first started infecting humans. And as people know, it started in 1996 in Hong Kong. First people got infected, and several died in Hong Kong, got infected at live bird markets. They were able to clean up, get rid of the live bird markets, slow down the H5 outbreak, but it reemerged in 2002. And since then, those goose Guangdong lineage viruses have continued to circulate and are really endemic in many areas of the world. At the same time, we start seeing other strains of influenza virus, not necessarily highly pathogenic avian influenza virus, emerging in people. We've got H9, H7s, we'll talk about more, and you can see number of cases over the years. And at this point, H5, H7, and H9 commonly accepted as being human infections, but we've also seen H10, H6, H4, and H11. A lot of that is through improved surveillance, but it may also be that there's just so much virus out there in the community and there's so much resorting happening that we're going to see more of these zoonotic events. Fortunately, none of them have been highly transmissible. And that has been, I think, the biggest thing that's prevented any of these from becoming a pandemic, because none of us will have antibodies to these, or maybe some T-cell immunity, but we won't have antibodies. So your risk factors for getting an avian influenza infection is very, very close contact with poultry, especially sick poultry. Certain jobs, processing those poultry, cleaning markets, increase your risk of getting infected. There's some really interesting data out of Vietnam and Cambodia where kids that swim or bathe in ponds that are heavily contaminated with avian influenza viruses have gotten infected. And the other thing is especially with H5, it appears that the people that are more likely to get susceptible or more susceptible to getting infected, getting severe disease, have particular genetic polymorphisms, especially changes in their interferon system. So there are some risk factors associated with getting an avian influenza virus infection. So let's take a look at what's happening with H5. It's really hard to find an updated map. This one is from 2018, but unfortunately it's not much different. H5 viruses continue to move westward. They are now endemic in many parts of Africa, the Middle East, and really throughout Europe. They're everywhere. You've got both low path and you've got highly pathogenic avian influenza virus. And low path is very difficult to deal with because you don't know your birds are infected. This is some recent data showing the country, the bird, and the genetic clade of H5. And you can see every country from Afghanistan, Bangladesh, Cambodia, Norway, in the 6-month window when we were doing surveillance, H5 infections have been identified in wild birds or poultry. And we continue to have human infections. And we've gone from calling it H5N1 to just H5, because what you see is that there are a number of different HA-NA combinations that can infect humans. And so this is from WHO, from a more recent strain selection meeting. And these are the number of people that have been infected with H5 in avian influenza viruses within a 6-month window. And it's Russia, Laos, India, Algeria or Nigeria, and China. So these viruses, as you can see here, H5N1, H5N6, H5N2s, continue to cross from birds directly into people, as well as other mammals, including marine mammals, and more recently, fox, I think that was in England. Unfortunately, these viruses continue to be very severe and often fatal when they do infect people. It can be younger people. It's, here you can see it's a 5-year-old, it's, a 52-year-old. So it's not the very, very old that we're used to. It's really what would be healthy adults, unfortunately. The H7 viruses gave us quite a scare in 2013. So this is a map of H7 avian influenza outbreaks between 2005 and 2017. And you can see a very wide distribution, much more so than H5. We've had H7N9, H7N8, H7N2s, ongoing problems in Mexico, both low pathogenic and high pathogenic. But we had these ennui lineages emerge in 2013. And at first, these were people exposed to birds in China in live bird markets. These were low pathogenic avian influenza viruses, so that means these birds had absolutely no clinical symptoms, and people were getting sick. Some were dying. It could be mild to severe disease. But what was concerning is that we saw transmission. There was some limited transmission in family clusters. So when we saw this and we had a lot of cases, watched very carefully, and what you could see is we had these yearly outbreaks, with 2017 really being very high load. And these were more people, if you think about it, than ever infected with H5 influenza viruses. So we were on a state of alert, basically. There were pandemic preparedness vaccine stocks, candidate vaccine stocks made and stockpiled in many places around the world, because we weren't sure if this virus was going to emerge and become that next pandemic. But since 2017, we've seen basically very few cases. So this is from the recent WHO meeting. It's absolutely no H7N9s seen in people. There was a very small outbreak of H7N2 actually in New York in shelters in cats. So cats got avian H7N2 infections. We have no idea where those cats picked it up because they had not seen H7N2 in New York before. And these cats were actually able to transmit to other cats, but also to several of the shelter workers, including a veterinarian. So this was a very small outbreak. We don't know much about the origins of this virus, and it just came and it went. It's sort of like that constant warning that you can't forget about these avian influenza viruses. Now, H9N2 is one that has a very, very wide geographic distribution. And you can see one of the first ones to actually show up in South America. So you can see it continues, again, to spread many times. Especially in certain countries like Egypt, you will have birds co-infected with H9 and H5 at the same time. And you can see how these strains have really exploded since about 1995. About that same time that H5, the Guangdong lineage, took off. And you can see these different colors are different countries, and just the amount of isolates. We're finding a lot in Bangladesh, in Egypt, and other parts of the world. There's a lot of genetic diversity with, 3 distinct really clades that are circulating, and many of which, especially this G1 lineage, are known to infect people. This is, again, from strain selection meeting from WHO. This is within a 6-month window. And you can see the sheer number of people, primarily kids in this case. H9 is really something that seems to infect children. All exposure to poultry, all very mild, still no person-to-person spread, but continual exposure of people to these avian influenza viruses. So this is ongoing. And again, it's not limited to these big 3. We've got other ones that are emerging and spreading. Some of them do have internal gene components from some of these viruses that may help explain their ability to cross that species barrier. But we still don't know what is that key viral signature needed for a virus to transmit between a bird and a person. So this, I think, makes me a little bit more nervous, is that this is not just avian influenza viruses. This is what's happening in the pig population. And these are the number of people infected with variant H1N1 viruses. So you can see this is throughout Germany, the Netherlands, Canada, so Brazil had several infections. And what you can see, this is the genome constellation of variant H1N1 or H1 viruses in Europe and Asia. And we have a completely different constellation of viruses in the Americas, where these are mixtures of classical swine, North American lineage viruses, avian, human, Eurasian, just continual mixing and matching. This has really happened since the triple reassortant cassette that sort of seeded the 2009 pandemic. Got into the swine population in the 90s. The diversity of swine influenza viruses has exploded. So this is variant H1N1, and this is variant H3N2, which are also co-circulating with these H1N1s and are again crossing into people quite frequently, typically children, close contact with pigs, mild disease in both the pigs and the people. But again, you know, these are of concern, pandemic concern, as they continue to circulate around the world. So one thing that I noticed when I came to St. Jude and got involved with both WHO and more of this global surveillance is if you look at any of these maps, whether it's H5, H7, H9, even human, you know, South America and really the Caribbean, it never is well represented. There's not much information. So doing a dive into the literature, when we really started looking at this in 2013, 2014, at that time, there were only 18 different hemagglutinin sequences of South American avian influenza virus in GenBank. And you can see that's 0.1% of all. You know, and you start looking at the history, there was a report in 2001 in Bolivia, and then you had an outbreak in Chile. And really, it was very clear that South America was, you know, an underrepresented or even a neglected area of surveillance. We know more about what's happening with flu in Africa than we do in South America. You know, much of the work that's been done is just beautiful work from Daniel Perez's group. And he really highlighted that what was known about avian influenza, especially in the wild birds in South America, is that there appeared to be two different lineages genetically, a North American lineage and a South American lineage. And they're really believed to be limited genetic exchange between the northern and southern hemisphere, which really doesn't make sense if you think about it, because if you look all around the world, you look in North America, look at South America is a really unique country in that there is a convergence of all of the animal or all of the wild bird flyways. So you've got the Pacific American, you've got the Atlantic, the Mississippi, all of them converge in some parts of South America, particularly when you get down here into Chile and Argentina. So we undertook studies, built great collaborations with what's now basically family in Colombia and Chile, and really wanted to investigate what was happening in the avian population. And we start by showing you some of our work in Colombia. And it's sort of also, I think, a cautionary tale. So, you know, the original goal was to characterize avian and swine influenza viruses through risk-based active surveillance on farms, in abattoirs or slaughterhouses, live animal markets, in wintering grounds. We, of course, got permission from ministries of agriculture and environment and, you know, the animal producing or production groups, the wild bird groups. And they were very excited to have us come in and start doing surveillance until we started finding viruses. And unfortunately, some of the first viruses we found in Via Vicentia were low pathogenic avian H5 viruses from whistling ducks. And we characterized those and demonstrated that, you know, these are just typical wild bird viruses. They're coming from North America, so these are migratory birds coming in, sharing their viruses, their genes. They were not, they could get into poultry, but they weren't a risk to poultry. But it's still H5 viruses. We then found H11 viruses, and these were spillovers from wild birds that got into some guinea fowl that were in live bird markets in some of the major cities in Colombia. It was tricky because about that time, Colombia had announced that they were avian influenza virus free, both in poultry and wild bird populations. So we basically were not allowed to do surveillance anymore. Just because of influenza is tricky. It's a very political disease. So you can see where this could cause problems. So at the same time, we were looking for swine influenza viruses, working with the swine producers. We found that it was very much endemic in many of the swine. And prior to 2010, we were able to show through some repository samples that really what they had was classical swine, H1 and H3 viruses. But then post the pandemic, all these pigs were getting basically a reverse zoonosis and were getting infected most likely from handlers and farmers. And that continued till about 2015, 2016, where we started to see some of the H3 came back. But they started vaccinating much more of the farmers, which was good because what we were able to show doing these animal-human interface studies is when we looked in swine workers, we'd look for antibody titers. So this was going on the farms, doing passive surveillance, so swabbing the people, swabbing the pigs every month unless somebody had a fever, influenza-like illness, looking for seasonal flu, which we could clearly show they had antibodies to seasonal flu, whether it be H1, H3, or influenza B. But they also had antibodies to different swine flus. And the more time and more contact they had with swine, the more likely they were to have titers. And again, this data was welcome in some ways, but in some ways not welcome. And so they started developing vaccine policies for swine workers. And we had to stop these studies in 2018. But we've been able to continue some really interesting animal-human interface studies in the Santa Marta, which is the Caribbean region of Colombia, where we're actually looking in rural and indigenous populations. So we've been given permission to look for influenza in these communities, and we got into it because when we went to visit a collaborator, the tribe said their ducks and their chickens were dying and the kids were getting sick that were in contact with them. Could we help them? So we have been working with them since 2016, and we have a lot of very interesting data. Now we're doing, obviously, SARS-CoV-2 in the communities, but we're able to show that clearly influenza virus, seasonal humans, goes through the communities, but depending on the tribe and their different genetic groups, they may or may not make good neutralizing antibodies to the virus. Their animals are often positive, and these groups, in many cases, will have higher antibody loads to swine and avian viruses than they will humans. So it's a really interesting community to look at the animal-human interface and start understanding which viruses are successful at transmitting and what is it about the population. This is a completely unvaccinated population, so it allows us to really start to understand also the immune response to natural infection. So let's turn to our studies in Chile, which have been incredibly successful. So it really started in about 2015. It was with Chris Hamilton West, who is an amazing veterinary epidemiologist at University of Chile Veterinary School. Pedro Jimenez Bloom, who was a postdoc with me, went back to Chile, and Nicholas Bravo, who was Chris's graduate student and then did a postdoc with me. But what Chris was able to do was to set up risk-based surveillance sites. So by looking at waterways, migratory bird routes, he was able to identify, I think it was 60 different sites, and we had to narrow that down But we look at the very top of Chile, central Chile around Santiago, and then down in Punta Serenas, which is down in that very southern peninsula. You know, what we knew was that there had been a high path outbreak in 2002 from commercial poultry. There had been some wild birds positive in 2007, 2009. And again, the government reported them AIV-free in 2010. So what I can tell you is that this has been amazing. There is a great diversity of avian influenza viruses in wild birds in Chile. So this first graph is blue is the number of samples, orange is the prevalence, and this is going from 2015 to 2021, and it's different months. So we're doing monthly or even every other month sampling, and you can see what it looks like. 2019 to 2021, you can see that the number of samples we've been able to collect has dropped off. 2019 in the beginning was civil unrest throughout Chile, and the rest has been pandemic related. But if you look down here and you look more at season, you can see that we get these seasonal spikes, typically in the spring and summer in Chile, of when we see increased amounts of avian influenza in wild birds. And this is typically when the migratory birds come in. It's when you have hatchlings. And you can see here in 2021, even though we're not collecting as many samples, we know what our hotspots are. And you can see that our prevalence has increased substantially. And this is actually by year. And again, the beginning, when we really got to, we were sampling everywhere, all of our potential risk sites. And then we got much smarter as we know where our hot and our cold spots are. So far, we've been able to isolate 217 different viruses, and we've sequenced many, many more. And what you can see is if you look at the hemagglutinin strains versus neuraminidase strains, we basically found everything except for HA, H14s, and H15s. Everything else is represented, including, and here's where you can see, these are actually pictures from some of our surveillance sites. It's amazing where we're finding. So all of our H9s are up here in northern Chile, around Santiago, especially in these areas where there's marshes, there's backyard production system. This is where we find a whole lot of virus. And then in southern Chile, it's also very different. We can find viruses, typically we find them in penguins as well. Here are our reservoirs, lots of wild waterfowl species, and you can see this yellow-billed pintail, the diversity we find in these birds. So this is another thing that was different about our surveillance. The rest of the surveillance around the world is very focused on particular bird populations. So domestic poultry, which makes sense. Wild birds, people will focus on blue-winged teals or mallard ducks or very specific bird populations where we didn't care. Whatever, and all we do is environmental sampling. So we will pick up a little bit of feces, fresh feces from the environment, and then we can do mitochondrial DNA to identify the bird species. We don't care what birds are there. We just care if they have flu. And this has allowed us to really better understand the reservoir and see how diverse this is. You know, we have these wading ducks, and then lots and lots of shorebirds, H13s, lots of gulls. So this has not only expanded our understanding of avian influenza in Chile, but also which bird populations, both migratory and resident, are going to get infected. And this allows us to pick now spots and bird species that we know we will successfully find flu. Great. All right, so the other thing we found that was quite interesting is most of the prior sequencing had shown just South American lineage. What we found is that many of our viruses were a reassortment. So they were a mix of North American lineage, shown here in red, South American lineage, shown in blue, some Eurasian, actually shown in gray, and then equine. And I'll show you more about that. So it's a really diverse mixture of genes that are in these bird populations. And what we found really interesting is it seems to have a latitudinal response, meaning that these viruses, if you look at what percentage of the genes are South American lineage, our strains in Colombia are completely North American. A strain that has been found in a couple in Brazil, northern Brazil, were completely North American. Then we get down here in Peru, we get a little bit of South American. Bolivia, Chile, it's about half and half. And then when you get down here in southern Chile and Argentina, completely South American. So it's really intriguing. We can see and almost predict these hotspots where we're going to get genomic reassortance occurring in these bird populations, really about here. We'd love to look on the eastern side and see what is happening in Brazil. Is it something very similar in Brazil as what we're seeing on this west coast of South America? So when we get these viruses, the first thing we really have to do is do some pandemic preparedness. We have to understand, especially these high risk, sort of our scary strains. So we put them through a virus characterization pipeline to ask, is it risk to specific groups like poultry or swine? Is there a risk to humans? We look at things like molecular determinants of virulence, receptor binding specificity, transmission. We can feed all this data into algorithms developed by either Centers for Disease Control or WHO that predict risk. And you can come up with something like this where you look at the risk of it emerging versus how big of an impact it would be if it emerged. And you use that information to decide what should be made into a vaccine. But we also use it to remain compliant and provide this information to both WHO as well as the local authorities. So when you find these kinds of strains in H5 and H7 and H9, you have to ask, is there a risk to these backyard production systems? Because that is a major part of animal production in Chile. There are these backyard farms where they have pigs and wild birds and chickens. And so these are our backyard production systems. And we did both serological as well as virological testing. And you can see these red dots are positive backyard production systems. And these are the strains that we were able to find. And we did tell the authorities. And what ended up happening is in about 2018, I think, they had a low pathogenic avian flu H7N6 outbreak in commercial turkeys in Chile. And because of our surveillance, we were able to show, basically identify majority of the genomes where they came from, because we had isolated this H7N6 and this H12N5 from these yellow-billed pintails or teals that we know contributed the polymerase acidic unit, the hemagglutinin unit, basically everything, but we don't know where the NS and 2 of the polymerase basic subunits came from. So this is the power of doing this active surveillance. The other thing that we've been able to capture is some interesting work on equine influenza viruses. So equine H7s sort of emerged, died off. The H3 equine viruses really showed up in the 60s, first in horses in Miami, but those horses had been brought up from South America. And we knew they had some avian genes, but we didn't know where are they coming from. And this is a lineage that now circulates both in horses as well as what gave emergence to canine influenza virus. And through our surveillance, we were able to find H3 viruses, there were 6 of them, all throughout sort of the central Chile that we thought were so intriguing when we sequenced them. We thought we were doing something wrong because this is showing the hemagglutinin phylogenetic tree. Here's where all of your wild bird H3 viruses are. Here's your equine, and here are our Chilean H3s. And we thought, well, that's really interesting. And through a lot of phylogenetic analysis with much smarter people than me, what we found is at the DNA phylogeny of avian H3s look like this. Here's your equine, and here's your Chilean H3s where they're an intermediate. And what we were able to find is really HA, NP, PA, and NS genes in wild birds in Chile that are really sort of the origin of these equine influenza viruses, which we thought were really exciting. Right now, we're characterizing some H13 viruses, which we found in gulls on the shore. I have no interest. Nobody's interested in H13 viruses unless they're interested in seagulls. But sequencing them, there are about 4 of them that we found have all of these molecular determinants of virulence that suggest that they could infect mammals. They can bind to mammalian receptors. They can evade MX and antiviral responses. What we actually think, so they will infect, a couple of strains will infect mice, will cause disease in mice. They will infect a ferret, although they do not transmit. What we're wondering is if they weren't originally found in marine mammals and then crossed into the wild birds. So now we're looking at sera from marine mammals around the world to see if these mammals have this H13 virus. Swine, lots of swine influenza virus in Chile as well. They have some, like 3 very unique clades of swine influenza circulating, one of which is an H1N2 that is a totally human virus, and that the HA is identical to seasonal H1 that circulated in the early mid-90s, and the NA was a seasonal one circulating before that. So people born after about 1980 would be susceptible to getting this infection. It transmits beautifully in ferrets, but people born, as you can see here, after 1990 have very little antibodies against this particular virus. We found lots of other things. We found avian influenza getting into dogs and cats, and I think it just shows the power of doing active, consistent surveillance, and we've been fortunate to have the funds to do that, to really understand the ecology of influenza virus in a country. And so with that, I think the big message is South America continues to be understudied. Every time they bring up funding for influenza studies and they want to go to Southeast Asia, they want to do Europe, I push and push for South America, because there's so much interesting work that needs to be done, both surveillance and basic fundamental research to really understand the ecology of influenza viruses. And I strongly believe until we understand what's happening in South America, we're not going to get a big picture of how influenza virus really crosses hemispheres and crosses species. And with that, I'd like to thank all of you, my team, funding sources, and especially all of my people that are on the ground. We've done a lot of capacity building. Most of the work gets done by them in their laboratories, and I just think that is so valuable. It's not viruses and things coming to us. It is them having the capacity to do everything, or as much as possible, themselves, which is absolutely critical for, continuing to have great research everywhere. And with that, I thank you as well, and I'm happy to answer any questions.
Well, Dr. Stacey, thank you very much for this brilliant talk. It was really, really nice and very interesting. I have a lot of people in the chat saying that the talk was amazing, so congratulations for that.
Thank you.
And I have a question for you. So you showed all these maps from avian influenza around the world, and we have this blackout in South America. And now you are working with Chile and Colombia and start to find some avian influenza. So what would be your guess about Brazil's situation? We have few studies that we have detection of H1, H3, H11, H9, but why we cannot find the H5 or H7 here in Brazil?
Yeah, it's a wonderful question. I wish we could better predict why. And we think about that a lot, is why haven't those high path H5s made it to the Americas? They're right there. Some of it could be the distance that they have to travel. Some of it could be the bird species. But why haven't you found it in Brazil? Well, hopefully it's because you don't have any. That would be the best scenario. You know, some of it is also how you set up your surveillance. I think having somebody like Chris Hamilton West, who is just an amazing veterinary epidemiologist and sort of risk modeler, having him be able to work like with Google Maps and weather and looking at environmental conditions and predict sites, and then having funds to go into these sites and start doing consistent surveillance. That's the only way we would have found it, because if you just go out and do surveillance once a year or twice a year and you don't know when to look, you may not find anything, which is what happens in many countries. Is, you know, it's expensive and it's time consuming and labor intensive. Going to these indigenous tribes to work with them, you know, those are amazing adventures every time we do it. And the same is true when you go out in the field. I have great pictures of some of the people swimming to get to where the birds are, or going in a boat. So I think it's being persistent, knowing where to look, and then when you start finding things, really characterizing what they are and what do you have. Wild bird surveillance can be very, very easy. You don't have to handle the birds. We have found that simply going to an area where there are many birds and just picking up fresh species from the environment is all you need to do. And now that we have primers for the mitochondrial DNA, we can identify the species of bird, which really makes a big difference. So we would love to come down and work with people, or have the people on the ground in South America come over and just start collaborating and see what we could find, especially in the times where we know it's in Chile. Those seasons, parts of the season, is it the same in Brazil, or is it completely different because you're on a different flyway?
Eurico? Thank you.
Thanks, Elena. Stacey, it was a great talk. Excellent. I have a double question. Considering the migration of birds all the way from the northern hemisphere to the south, to the tip of South America, how do you explain this sort of equatorial divide between north and south? This is one question. And the other is, are those mutations that enable binding of the hemagglutinin to alpha-2,6 galactose sialic acid also in the same region or nearby in the H7s and H9s that we should be scared of for spillovers to mankind?
Yeah, so two great questions. Let's start with the first one, is why is there this divide? Probably part of it is migration, right? So when some of these birds, so we know, like in the US, if you look at people that study the Mississippi flyway, these blue-winged teals, they go down and they migrate in Colombia. And I think what ends up happening is some of those, so you have the long-distance migratory birds, but you have the shorter distance. So I think you've got those long-distance ones coming down and then seeding into shorter distance migrators that start moving it down. You can almost imagine, like if I was going to get in my car and drive from here to Wisconsin, I would stop along the way and leave different things. And I think you've got ones coming straight from North America, stopping in northern South America, Colombia, mingling with some that continue to go down, and then they just spread locally. That is the only way that we can really imagine how it happens. But it looks like Peru, Ecuador, northern Chile, even around Santiago, there seems to be an awful lot of mixing in those areas. That's where we start to see almost every virus is a reassortment between northern and southern lineage genes, which I think is just intriguing. Second question about the receptor binding specificity. I think your question was about the H7s and the H5s. Do they have them? Yeah, some of them do. And I think, there were some very controversial studies in about 2012 from two groups, one in the Netherlands and one in the US, where they took that high PathH5 and passaged it, or made mutations to see how many passages would it take before that virus could now transmit effectively in ferrets, which is sort of our model for a human. And it didn't take very many passages before that change occurred in those receptor binding sites. Wow. So that is something we always look for, and why with that H13 virus that we're characterizing, we would never think to characterize an H13 virus, but we sequenced it. And based on sequence, this will bind better to a mammalian receptor than it will an avian receptor. And that's exactly what we found.
Thank you. Thanks, Flavio and Elena, for having me backstage. It's nice.
Sure. So, Dr. Stacey, I have a comment. Actually, it's not a question, because it was a fantastic talk. But I was impressed by one thing that you said at the very beginning of your talk about the detection of human influenza during the pandemics, which was a flat line. And we had an experience like it happened in the US as well. But the universities, they made an effort to help the official laboratories in diagnosing COVID-19. But we are adapted to do only COVID-19 diagnosis. Every time that we have a sample that is negative, we're supposed to send them back to the reference laboratories for them to be tested for influenza, which never happened because the official laboratories were so overwhelmed with samples to do COVID-19 that they were never doing the other respiratory disease diagnostics during this time. And I don't know if it has improved it at all.
It's, it's so hard right now. Everybody is exhausted from doing this, because we also had to set up employee testing and screening everybody. Supplies, it's very difficult to get supplies to do testing or even basic research. And so, being able to just stay ahead of SARS, it's understandable where flu would sort of fall by the wayside and people may go back and test them later. But yeah, we see the same thing here where people are testing for both. We're actually now in our indigenous cohort where we have a cohort now where we're specifically watching SARS. We're enrolling people that diagnosed positive with SARS, and then we're monitoring them, making sure that everybody, gets the treatment that they need. But we just did a test yesterday that it seems that the human seasonal coronaviruses are co-circulating in that population. Yeah. And so it's not something anybody's looking for, because again, we're just trying to keep ahead of SARS. That might be one of the few good things that comes out of this awful pandemic, is that it really has allowed academics to work closely with public health, and not just help with testing, but to help with sequencing. So, the academic institutions in many places were the ones doing all the sequencing of SARS-CoV-2 and putting it into publicly accessible databases. And if that were to continue, you could imagine for influenza or antibiotic-resistant bacteria or even other seasonal viruses and pathogens, it may change. I'd like to hear what Eurico thinks about this. It could change what we know about local sort of outbreaks and disease circulating within communities.
Yeah, it's interesting. It has affected even a disease that are not related to respiratory disease, like dengue here. We were seeing a decrease in detection, but we're not sure if this decrease is due to the fact that people is not going to the hospitals because they're afraid to go in there and get tested because of all the environment, or we're just missing that information because we're so focused in something else. Well, we will know someday. Yeah,
no, it's amazing that you're right. Everybody, while SARS is circulating, we're seeing, less of seasonal flu. People are having less allergies, could be wearing a mask. Now, RSV has spiked again. I don't know if you're seeing it there, but in the US, And in Colombia, RSV is going way up. So something is changing. It could be the Delta variant allows more, or people are tired of wearing masks and quarantining. But it's going to be really amazing to look back. So now we have all these samples, and if the time ever comes, you can go back to that repository, hopefully, and say what really was happening during that time. But you're right, there are people getting dengue that are not going to go to the hospital. You don't want to go in there and get SARS. Yeah.
Exactly. So, Ellen, is there any other question there?
Yes, we have a question. So Dr. José Luis Modena, he also congratulates you for the presentation, and he asks, do you know how RNA interactions can act to favor some type of genetic resortments?
Yeah, it's a wonderful, wonderful question. I think that's one of those really critical areas where we need more people doing that research because we don't understand it. There's going to be two things. One is that right combination of genome segments, and the other might be somewhat of the host, getting a host. That's why we look at people that are vulnerable, because they're more likely to get infected. They have higher viral titers. They may allow more reassortance. But we do know that there's something very unique about the 2009 pandemic. Those internal gene segments were very promiscuous, meaning they would allow a lot of different HA and NA genes to interact and to actually form productive virus. So there was something unique about those internal components of the polymerases and the nuclear protein that allowed so much recombination to occur. And we're using that as sort of that, this is something we know will reassort. Let's understand what it is about that allows it. And it is going to be a combination of things, but NP and the matrix protein seem to be very important in getting those genome segments together. It's a great question.
I have one comment and another question. So when you said that maybe the lack of detection here in Brazil could be how we set up the virus surveillance, one comment was more about when we had the Chile case of the H7, was really surprised for me because they had almost 0 or 2% of mortality in the flock. Mm-hmm. And the Brazilian surveillance here, it's set up in another way because they have, the flock needs to have a very high mortality to have that flock to be screened for avian influenza. So at that point, we see that we can miss a lot of cases here. And another thing that was really surprised that this virus was positive for the real-time based on matrix gene, but was negative for the H7 test. Mm-hmm. And then they proceed to do the sequencing even though it was negative for H7. And here, the reference laboratory, they just don't go further. So it's negative for H7, then they just stop to look for it.
Yeah.
So I think that can be one reason. And my question for you is, when you show all the detection in Chile over the years, could you see the same pattern that we see in Europe when we see the changes of the subtypes for the HA's group? So you switch the H1 and one other year you can detect only H3. So we see that difference. Did you see that in Chile as well?
Yeah, that's a great question. That's something we're really looking at now. Now that we have, so many years of data, we can actually go in now. We have enough data to go in and say, okay, how did this change by the year, even by the site? And so we're just starting to do that now to really try and understand, okay, if this site we only see H1 and H5, why is that? So then you can say, well, what are the bird species there? And so we're now really starting to look at that. And actually we're working with several mathematical modelers that are starting to model all of these for us to even say, how are these things? So if a bird comes into the site, we know it has H7. Based on our data, what is likely to happen? So it is a great question and something that we are doing because it's also going to help us moving forward decide which sites do we want to focus on, because every year there's less money. So every year we have to get smarter at how we're doing this. And we're always happy to share any of the reagents that whether they're viruses, whether it is sequences, whether it's ferret anti-CEIRR, any of that, we are always happy to share. So if that is useful to anybody that you know, we would be thrilled to send whatever you would need. Primer probes that we use, whatever you need, because we just think it's so important that, you know, we do as much in those local areas. And if part of it is, okay, we don't have the reagents to look for H7 or well, we do, and we'll send them to you.
Nice, thank you. And last question, how did you get so many samples? I saw like 5,000 per season in Chile. How did you get there?
Oh the team there is fantastic. They are so good, and it's just this wonderful combination of veterinary students, of graduate students, even local people that are just very interested in birds that want to help. And so what they will do is in their sites, they've identified sites that they go to monthly. They have sites that they go to every 3 months or every 6 months. And then they have some sites that they even go to more frequently because they know that they're going to be positive. So they have the teams that will go out and collect the samples. And again, with the wild birds, it's very easy in that you know where to go. There's all these waterways, and you just go and you pick up poop and walk around, and it's beautiful there. And it's really, it's just worked amazingly well. And then they can pool samples for testing, which makes things much easier. And we are now getting set up to do MinION sequencing on site. And rather than having to send it to us to do full genome sequencing, so they can do a lot of the sequencing themselves as well. So part of that capacity building. But yeah, they are amazing. And moving forward with our new contract, they're looking a lot more in the swine populations, and they have it where they know when the swine are there, they know when they should be positive, they know when those wild birds are going to be in those areas, and they're actually setting up video cameras in these backyard farms to watch interactions between all the different species, which I think will be really intriguing.
Wow, yeah. Today I saw a video that was a bat interacting with some of different species as well, some deer here in Brazil as well. So things that we never think about, but yes, in the nature, with the wild life, everything happens.
Yeah, especially at water sources. Everything will go to a water source.
Yeah. So I don't have another questions in the audience. Eurico?
No, I have no additional questions. Just I'd like to thank Stacey again for the great talk.
No, thank you so much for the invitation. I was so happy to get it. So I hope it was useful.
Wow, it was definitely useful.
There's certainly was.
Yeah, there was a lot of people thanking you at the comments, and I would like to join them and thank you again. And in the name of all,
we have a last question.
Oh, is there one more question?
Yeah, last question. So yeah, it's here. Can you read it?
I can read it. Yeah. So that is also a wonderful question. You have lots of people that should come work on flu with us because they ask exactly the same questions that we do. So I think that it's really going to be that closer contact of humans with those farm animals, the domestic poultry, the pigs, much more so than the migratory birds, because we still have not seen a direct transmission of a wholly wild bird virus into people. the few times that it's happened, it gives you like pink eye, conjunctivitis, but no infections. It always is coming from a domestic poultry or a mammal of some kind. So I think those are the much bigger risk. Having said that, in Chile, we're going to start getting just blood samples from fishermen that are in this area where that H13 virus is, and we'll see how many of them have antibodies to some of these avian viruses. And if they do, it means that they've been exposed to the virus. It doesn't mean they were infected, but they had enough of an exposure to have an antibody response. So I say that we have no evidence, but perhaps we haven't looked. So stay tuned. We'll let you know what we find.
Wonderful. Thank you very much for your talk, the discussion as well. It was very, very interesting, and congratulations for your work.
Thank you, and thank you so much for the opportunity. I really appreciate it.
Thank you, Dr. Stacey. I'll switch Portuguese now, just to say goodbye to our audience here. Pessoal, muito obrigado para quem esteve com a gente nesse tempo todo até agora. Né? Então, a gente finaliza essa edição do SBV Talks de setembro. E agora o próximo a gente espera vocês no Congresso Brasileiro de Virologia, número 32. É 32, Helena? Me ajuda, 32. Às vezes eu perco a conta. Mas, então, no próximo, daqui a 19 dias, a gente inicia nosso congresso e a gente se vê lá. Muito obrigado a todos pela audiência, pelas perguntas, e a gente se vê em breve. Bye, bye, all.
Bye, thank you.
Bye.